Hello everyone, I'm Fien Wagenaer, working at Ghent University at the research group CMET, and I'm focusing on drinking water microbiology. Today I will tell you something more about drinking water biofilms. As you might know, we have first the drinking water sources, such as surface water for example, and those are treated in the treatment centers by the drinking water providers and going via drinking water distribution systems to you and me, so the customers. The drinking water providers can guarantee safe and qualitative drinking water when the water leaves the treatment centers, but during distribution it is more difficult to control this quality. For example, your water quality can change because of interactions with the pipes and the biofilms. Organic compounds can be transformed. There could be microbial regrowths and so on. But you should know that there are a lot of parameters that are controlled and strict requirements need to be fulfilled to distribute tap water. The most important water quality parameter for us, and where we will focus on today, is total coliforms. It's an indicator parameter, so the coliforms themselves are not always harmful, but they indicate that there is something wrong with your water quality. In Flanders, there are still norm exceedances of these parameters. The source of contamination is mostly unknown, and we know that the coliforms have difficulties to survive in the oligotrophic environment. So one of our hypotheses is that they could hide in the drinking water biofilm on the pipes. As drinking water pipes are located under the ground, it's challenging to sample drinking water biofilms. Therefore, you used a biofilm monitor to grow and sample the drinking water biofilm. Water is following from on the picture here to here, so following the arrows, and the monitor consists of glass rings to collect the biofilm. We studied the biofilms at two different places. One was receiving treated groundwater without the disinfection step, and the other was receiving treated surface water with a chlorine residual. We let the biofilms grow for 1.5 years to have a mature biofilm. Confocal laser scanning microscopy is one of the techniques that we've used to study the biofilms. With this technique, we can get high-resolution 3D scans. First, we wanted to visualize and quantify the EPS content of the biofilms. So you have your biofilms on the pipes. In the biofilms there are bacteria and other microbes present, and they are then embedded in a layer of sugars and proteins to protect themselves from the outside stressors. And this sugar and protein content is called the extrapolymeric substances, or the EPS content. Secondly, we want to prove our hypothesis by performing invasion experiments with a GFP-labeled coliform Serratia fonticola. So the biofilm rings are not normal structures that you can easily put under the microscope, so we needed to build a kind of structure so that your biofilm could easily be stained and rest on a cover glass. As you can see here on the pictures, we made this kind of structure, and to target our different biofilm compounds, we use different dyes that bind with these compounds. For example, to stain the bacterial cells, we use DABI staining to target DNA. Cyperruby binds with the proteins present, and concanavalin A interacts with the sugars. These dyes have different excitation and emission wavelengths, as you can see. For example, the laser of 405 nanometers will excite the DABI stained cells, and we can see the signal in the region of around 500 nanometers. The stains did not interact with each other, and the right combinations of lasers and filter cubes were available at the Nikon microscope of the proeftuin. Here you can see the results for both biofilms. Cells are shown in blue, proteins in red and sugars in green. And there is also an overlay. You can see that the cells are interconnected with the EPS content, and we could also see that the biofilms formed under treated chlorinated surface water supply are less dense than the biofilms formed under groundwater supply. Next to visualization, we also wanted to learn actually from the scans, and using ImageJ and COMSTAT2, we were able to quantify different biofilm parameters, for example, biomass, roughness, and average thickness. And zooming in on, for example, the biomass content, we could see that the biomass for biofilms resulting from groundwater was higher. However, to perform the analysis with COMSTAT2, you always need a kind of a thresholding step to distinguish between signal and background. Here we use an automated threshold, the Otsus threshold, but there are a lot of other thresholding methods available, each giving slightly different results. As mentioned in the beginning, we hypothesized that coliforms can hide and settle into the drinking water biofilms. To investigate this, we labeled a coliform, Serratia fonticola, isolated from the distribution network in Flanders, and we labeled this coliform with a green fluorescent protein. We spiked the coliform in a vessel connected to the monitors, and after a couple of days we took out some rings to see if the GFP-labeled coliform could be detected. Important to mention, earlier studies performed invasion experiments with pathogens in high concentrations. Here we wanted to simulate the real-life network, and as the limit for coliforms is 0 coliforms per 100 ml, and if there is detection in the net, the amount that they detect is around 100 cells per 100 ml, so we wanted to spike on average this amount in our vessels. We also followed the behavior of the coliform in the bulk water, but we could see that after 24 hours, the coliforms could not have been detected anymore in the bulk water. Here you can see the results of one of our monitors. So in blue again, you see the DAPI stained cells. In green, the GFP labeled coliforms, and an overlay picture of both. But for this application, the technique confocal microscopy is difficult to quantify. So we also performed qPCR to detect if the green fluorescent gene was present, and as both techniques give a positive detection, we could conclude that Serratia fonticola was able to settle in the drinking water biofilms. To summarize, we used confocal microscopy to visualize and quantify the EPS content in drinking water biofilms. The dyes that we've used worked well, but it's difficult to choose the right thresholding method. In the next part, we showed that Serratia fonticola, a coliform, is able to settle in both drinking water biofilms. But what does this mean for practice? If there is contamination, the coliform will have difficulties to survive in the oligotrophic environments, but it can go to the biofilm and maybe contaminate the water again in some circumstances. This outcome is important for water quality management, and further research should focus on detachment strategies of the biofilms and of the coliforms. I would like to thank my team and you for listening.